Good morning, everyone. I want to first thank the organizers for inviting me to present. The first time I actually attended this workshop was 17 years ago in this very city of Amsterdam. So I feel like this is sort of a homecoming. What I'd like to do today is talk about some of the mechanisms that the Division of AIDS and the National Institute of Allergy and Infectious Diseases has to support pharmacology research. And many of you who are in this room have taken advantage of these opportunities and very actively participated. So it's going to sort of be preaching to the choir, but I think the overview will be helpful for many of you who aren't as familiar. And so I'll start with this slide. And this is describing the therapeutic research program, which I am part of in the Division of AIDS. And our mission statement involves improving HIV clinical outcomes by advancing translational, having a little trouble with this pointer, translational and clinical research. So the focus is both on basic, translational, clinical, and also implementation science research. And again, our interest primarily is in therapeutics of HIV and HIV-associated infections. One of the areas that we do a lot of work in is the possibility of a functional cure for HIV, and of course that involves a lot of pharmacology work. But as you can see here, novel, durable treatment strategies for HIV, also Hep C, Hep B. We're very much interested in long-acting products, novel formulations, but also new drug targets and molecules. So interest in, again, basic science, preclinical work as well. Now, a lot of our clinical trials are done through networks. We have a series of research networks in the Division of AIDS, and this turns out to be a really, really efficient way to do clinical trials. And these networks are individually funded and operate fairly independently. The oldest network that we have is the AIDS Clinical Trials Group, the ACTG. It actually was formerly known as the AIDS Clinical Trials Group, but I think the name has been changed to Advancing Clinical Trials Globally. We have the IMPACT Network, which focuses on maternal and pediatric HIV. We have the HIV Vaccine Trials Network, and I think that title speaks for itself. And then we have the HPTN, the HIV Prevention Trials Network. And pharmacologists, including many of you in this room, are involved as principal investigators of many of these studies, as well as serving as protocol pharmacologists. These networks have clinical trials units, and they also have research laboratories, and we literally have these labs and clinical trials units on every continent. They contain these transformative science groups. So these are networks of investigators, and they decide what the research agenda is going to be, and they also propose the actual clinical trials that are going to be conducted in the network. And so here's an example of one of these trials. This is ACTG 5357. The principal investigator is Dr. Babafemi Taiwo, formerly at Northwestern University. I think he has left and gone to ViiV. And so this is looking at combination injectable cabotegravir plus the broadly neutralizing antibody VRC07, 523 LS. So this is the long-acting modification of the broadly neutralizing antibody, and looking at its ability to maintain suppression in adults with HIV. So in this study, they took individuals who were already suppressed on antiretroviral therapy, and they switched them to the combination of these two long-acting products. And what you see here is a Kaplan-Meier curve looking at cumulative probability of confirmed virologic failure. And over a span of nearly a year, we see slight increases in virologic failure, but at the end of this period, the failure rate is 7.3%. Now, you could look at that the other way and say that there were over 92% of individuals who remained suppressed on this particular combination. And then in this study, on the right-hand panel, they were interested to see, well, what did the PK look like in these individuals? What were the drug concentrations? They looked at trough concentrations of cabotegravir and found that in all these individuals that failed, they still maintained plasma concentrations above protein-adjusted IC90s. And similarly, for the broadly neutralizing antibody, they found that the concentrations were above the target concentrations. So, why did these individuals fail? Maybe they had archived integrase resistance. Maybe they had baseline resistance to the broadly neutralizing antibody. Not exactly certain on the causes. I'll mention here that we also have the Clinical Pharmacology Quality Assurance Program, or the CPQA. Many of you are very familiar and participate with this. And the purpose of this particular contract, so this is a contract, one of our quality assurance contracts, it provides a comprehensive quality assessment program for clinical pharmacology laboratories that actually test the samples that are collected in these clinical trials through our networks. And again, it's an ISO accredited proficiency testing program. The proficiency testing program is one of the key services of this particular contract, but they also help labs in terms of validating their assays for the drugs that they're testing in their labs. Now, the NIH has a way of kind of catching the attention of researchers and trying to persuade them to do work that we feel is important or is a priority for us. And this is done through what we refer to as a RFA, or request for applications. And through this mechanism, we're saying, okay, you have the expertise, you know how to do this research, why don't you help us answer key questions or help us address certain research gaps. And this is an example of an RFA that I released several years ago, HIV drug resistance, genotype, phenotype, outcome correlations. So I was particularly interested in better understanding how different HIV subtypes may differ from subtype B in terms of the patterns of resistance mutations or the frequency of resistance mutations. Also, the fact that we often see discordance between genotype and phenotype and clinical outcomes. So what's going on here? And released this RFA and got some really excellent projects that were funded. And since we're in the Netherlands, here's an example of one of those. This is a grant that went to David van de Vizier at Erasmus University in the Netherlands. Novel mechanisms contributing to failure of dolutegravir-containing combination antiretroviral therapy in clinical practice. So the first aim of this particular study was to characterize the pattern of integrase resistance in individuals on dolutegravir. This is being done in Brazil And they're going to compare subtype B with subtype C, which they have some subtype C there. They also have subtype F, and they also have this peculiar BF recombinant. So they wanted to look at mutational patterns. They also were very interested in this 3' polypurine tract. And this is a region in the HIV genome. This is outside of the integrase gene, but we are probably familiar with the fact that mutations have been observed in this region that may contribute to integrase resistance. And then they want to look at the phenotypic consequences of the mutations that they found. And here is a figure from one of the papers they published. And in this corner, you can see the HIV genome. The polypurine tract is actually about 15 nucleotides that is purine rich. They're almost all purines. And so what they did is they generated a number of mutations in the polypurine tract. They transfected it into cell lines, and then they looked at the replication in these cell lines. This first set of bars, this is wild type replication in the dark bar, and the lighter bar is in the presence of dolutegravir. So you can see that the dolutegravir is reducing replication by probably close to about 80%. But you can look at the different mutants that they produce. The first thing you note is that their replication is reduced, so there seems to be an effect on replication capacity, but also the addition of dolutegravir has no effect on replication. So what we're seeing in vitro is what has been proposed, that these mutations can affect sensitivity to integrase inhibitors. Now, a lot of people have their own ideas on doing research. They have their own interests, and we have a different mechanism where they can apply for an R01 grant. And these we refer to as investigator-initiated. So you can come up with your own concept, you write a proposal. If this proposal matches our priorities, and has the potential to advance the field, then you can receive a grant. These are 5-year grants. Again, the R01 mechanism, meaning that it's hypothesis-driven and has very robust methodology and a high likelihood of generating some good data that can be valuable to the field. Here's an example of one that we just awarded. This grant went to Eli Rosen, who you'll be hearing from a little later today, at UNC Chapel Hill, Jake Estes at the Oregon Health Sciences Center. And the title of this particular project, Assessing Time to Treatment Variability in Antiretroviral Penetration by Multimodal Imaging. So what this group is interested in is the penetration of antiretroviral drugs, or how well do these drugs actually get into reservoir tissues in particular. And they're interested in long-acting drugs. Do these long-acting drugs better penetrate into reservoirs of interest? And they're using this really robust technology referred to as IR Maldesi MSI. And if you're curious about what that is, that's infrared matrix-assisted laser desorption electrospray ionization for mass spectrometry imaging. So thank goodness they call it IR Maldesi. They're using this novel method to look at the, again, penetration of the drugs into particularly lymph nodes, but other reservoir tissues in rhesus macaques. They're using other types of imaging technology, doing some in situ hybridization, transcriptomics, metabolomics. And just to give you an example of how robust this tool is, If you look at the left-hand panel, you can see the anatomy of a lymph node from a rhesus macaque. In the center panel, you see some of this imaging being used. You can look at CD4 cells, you can look at antiretroviral drugs, you can look at viral RNA, and you can superimpose these images and see where in the lymph node these different elements are located. You can actually map the distance, in this case, antiretroviral drugs, difference in the distance to where viral RNA. So you can use this tool, and you can see here some analysis they did on the graph looking at the distances. So this is just an example of how this tool can be used, and this is what they're starting their project. We're looking forward to getting some very good data from this study. Now, we also have investigator-initiated clinical trials, and these are done through this UO1 mechanism. And so I think Rodney Ho, who is one of the investigators on this particular project, he's certainly no stranger to this group. Rachel Bender-Ignacio was the principal investigator. Long-acting platform for targeted combination antiretrovirals. So what this group did is they took this novel formulation, it's a lipid nanoparticle, and they inserted 3 chemically different antiretrovirals into the same lipid nanoparticle. And they're interested in looking at the PK of this novel formulation. They're interested in the safety and tolerability of a single subcutaneous injection in the plasma. And of course, they have a number of exploratory objectives as well, including looking at the ability of this product to get into PBMCs. And so one of the issues that came up when we started this study is, how do we get enough product to do the study? If you look here, this cartoon illustrates kind of what this particle would look like. These are the actual phospholipids that are used to form the particle. And these are the 3 different drugs: lopinavir, ritonavir, and tenofovir. And as I mentioned, they have very different chemical properties, from being very hydrophobic to being hydrophilic. Yet we're able to put all 3 of them in the same lipid nanoparticle. This is what the product actually looks like. So when they came to us, they're like, well, we need some product to do the study. And we're like, okay. Well, we have another important component in our toolbox, and this is our preclinical contract. And this assists folks in academia, small companies, in terms of getting products through development phase to apply for investigational new drug applications. And we have different task areas. And you can see here preclinical pharmacology and toxicology, in vivo testing and screening, chemical synthesis, so we can actually help labs develop or refine products, small animal model testing, and then we use this particular task order, formulation development and manufacturing, to make the product that was going to be used in the clinical trial. And so these are the results, and in the blue we have 3 different doses of lopinavir, in the orange 3 different doses of ritonavir, and then in the purple 2 doses of tenofovir. These are plasma concentrations. In the orange, we see ritonavir very quickly leaves circulation in the plasma. tenofovir persists a little bit longer but doesn't do very well. But amazingly, and particularly the 2 highest doses of lopinavir remain undetectable. These are well above target concentrations for over 2 months. And keep in mind, there's no ritonavir in the plasma to boost it. The ritonavir is all gone. So this is, I think, pretty amazing. That was presented at CROI. And then most recently at CROI, they looked at concentrations in PBMCs. And in the red, you can see PBMC concentrations. In the blue, you can see it in the plasma. And we're looking at the 3 different doses for lopinavir, ritonavir, and tenofovir. And interestingly, even in drugs that don't show up very well in the plasma, you can see concentrations extending out in the PBMCs for all 3 of these drugs. The last slide I'll show is a separate mechanism. This is actually a resource, a R24 mechanism. I think many of you are familiar with the LEAP program. This is Charlie Flexner's grant at Johns Hopkins, long-acting extended release antiviral resource program. So this is an actual resource to help investigators develop long-acting formulations for viral infections. So this is my last slide, and if anyone has questions, maybe you can get the mic or come to the microphone, whatever, while I just quickly go through this. We have first the clinical trials networks, ACTG, Impact, etc. We can use RFAs for promoting research and things that we think are important. But we also have investigator-initiated research, both for translational and clinical science research. We have a preclinical contract that can help with drug development. And then we have resource grants like the LEAP, like the Stanford HIV Drug Resistance Database. And I think that is my last slide. Thank you very much for your attention. And if I have time, I'll take any questions.